Okay, a couple more things. So here we see a chart that is comparing the number of changes in pseudogenes versus protein coding sequences. So pseudogenes are DNA sequences that are no longer used, they are no longer expressed, and therefore it doesn't matter if you change the sequence because you are not going to end up in reality using that protein anyway. So it doesn't matter if you have synonymous mutations or non-synonymous mutations. And overall, you see more in general. And then if you compare that to synonymous substitutions and non-synonymous substitutions in a gene that is being used in the organism's lifetime, you see that synonymous substitutions are higher, they're more common than non-synonymous substitutions. And we could say that, we'll say that we're looking at things that are fixed in the population. So in other words, the entire population has this, this mutation, which is synonymous when compared to the ancestral sequence. And these synonymous substitutions are fairly common compared to non-synonymous substitutions because they don't change the protein. So again, they're not rampant because we still have mechanisms to prevent any mutation whatsoever, but they're much more common than these non-synonymous substitutions. So we see the non-synonymous substitutions, which will change the amino acid called for, and therefore are more likely to change the protein and the function of the protein. These are much less common. We see there's fewer than one per billion years that occurs for a certain number of base pairs in this particular gene. And these are much less common because they are selected against. So when you change a protein, you just put a random amino acid in there, substitute out an amino acid, is more likely than not going to make the protein less functional. So it's not going to be as good at its job. Maybe it'll just be slower. Maybe it won't be able to bind something as well as it used to, or perhaps it won't even be able to fold in the correct configuration at all. And so that's why these non-synonymous substitutions are so low, because usually that is deleterious or inhibitory to an organism that possesses them, and they will not reproduce and produce as many offspring as individuals that do not have that mutation. All right, now on to the next concept, gene duplication. So here we're moving away from SNPs. And this is a really important concept, that is the concept of gene duplication. And here we actually see an example of gene duplication that has happened in human history. And so these genes are for myoglobin, alpha chains, zeta chains, epsilon, gamma chains, delta, and beta. What are all of these? Well, these are protein sequences that are used for hemoglobin, which is an oxygen carrying molecule. And so myoglobin is not part of hemoglobin, but it is a very similar oxygen carrying molecule in humans as well. The alpha, zeta, epsilon, gamma, delta, beta, they are different variations of this protein that are used at different times in life. So for example, a fetus has different oxygen needs than an adult human. And so they are going to be expressing different proteins that do very, very similar jobs. And only at specific times in an organism's life are you going to see these different genes produced. But again, they have the same function. They bind to oxygen, bring it to deoxygenated tissue, as we're going to see in a couple of classes, and they release the oxygen there so that your cells have oxygen. What does the cell do with oxygen? Well, they are going to use that oxygen for cellular respiration. I'm not going to explain all that, but you can review it on my YouTube channel or from your textbook, whatever you choose. What this image is showing is that all of these proteins are very similar, not just in their shape and their function, but also in their DNA sequence. And this is most likely because we believe gene duplication events, so a coincidental duplication or resulting in 2 copies of a single gene in a genome, resulted in the use of both of those different copies separately. So it's kind of like, I think that there was an example of this on New Girl. I can't remember now, but no, I think this was a Reddit thing. Allow me to go on a tangent for a moment, because this is a really good example and it's amusing. So if you think about kitchen tongs, so like the things that you use to pick up like a whole bunch of lettuce, or that you use to turn things over on the grill. Someone had said, was I wrong in using these they had to pull something out of melted wax, and they used their roommate's kitchen tongs, and they didn't know what to do about it. So they used the tongs because they didn't want to get burned, obviously. And they said, you know, I washed them off and they're fine, but I feel like I should tell my roommate. What do I do? Well, this would be a case where you should get a duplication. We should have a duplication event. You should have a set of kitchen tongs that you use to dip into hot wax, and then a set that you use for food. And even though they might be the same exact brand and the same exact product, then it doesn't matter. So let me just pull an image here to make sure that you guys know what I'm talking about. And share again. So this is what I'm talking about, just these things. So you have one set that you use for whatever art projects you have that you need to dip into wax, and then you use one set that is for food. They might be identical. You might have this expensive set that's $40 from Williams Sonoma. You might have 2 of these, and they have 2 different purposes. Why am I going into this? Because you can actually have the same situation with gene sequences. And now that you've duplicated these gene sequences, you could use them for different things. One for an art project and one for food. I'm just kidding, because that's not what you use these genes for, these proteins for, but you can kind of get what I mean now, right? So once you have a duplicate of this specific gene, then you could start to change or accumulate mutations in one of those gene sequences, maintain that function, and you might have different mutations in the other gene sequence. And so this was one duplication event here. And perhaps we had more duplication events that cause these other variations of the proteins. But we know that these all have a common ancestor. They're all fairly similar in gene sequence and nucleotide sequence, and in their shape and their function. And this is what we think allows for similar proteins to kind of hone in, to zero in on their specific function. And so again, we said that myoglobin and hemoglobin are very similar, but they have slightly different functions. As we could see, hemoglobin is made up of 4 subunits, myoglobin is made up of 1. And so this will lead to them having slightly different functions, in particular because they have slightly different affinities for oxygen. In other words, they're more likely to bind oxygen at slightly different concentrations. And that allows myoglobin to be better at bringing oxygen to muscles, whereas hemoglobin might not be as good at bringing oxygen to muscles. And again, we'll learn more about that later in the class. The takeaway here is that when you have a gene duplication event, which likely occurs randomly, again, this could be an accident, something that happens due to mistakes on the part of DNA polymerase, or even really big events or DNA damage where you get not even UV, but X-ray damage or cosmic ray damage to DNA, and you have a double-stranded break, and then you accidentally duplicate a gene when you're repairing that DNA. That can actually result in a good outcome, because now you have 2 copies. You could use them for different things. On the other hand, you might also use the 2 copies for the same thing and just make way more of the product, way more of the protein than you previously could, because you have more access to that DNA. Okay, I think that's everything that I wanted to go over today. So as always, you can email me or make an appointment with me if you have questions.